All right, good morning. It is Monday, January 25th, and this week we're working through the lesson Chemical Basis of Life. So you're going to access this module through Canvas, and you want to load Chemical Basis of Life in a new window. Here, this is an auxiliary lesson, so it will auto-load into your gradebook. So for this, you do not need to write a one-sentence summary along with loading your scores because it should load automatically. All right, in this video, we're going to walk through, I'm actually just going to do the lesson. So you can do the lesson with me, and that allows you hopefully to move through this without any issues. So here we go. And learn a little bit more because I'll add my own narrative to it. So in this lesson, we are learning about the basic building blocks of matter. And we'll read here. At its deepest level, the basis of biology is chemistry, matter and energy interacting in complex patterns. To learn about the chemical basis of life, you'll explore the components of cellular structures and what these components are made of, blurring the line between matter and energy, chemistry and biology. So a lot of this might feel like chemistry to you, and that's because biology is built off chemical molecules. That's basically, I like to say, that's basically all we are. We're just a bag of chemicals, all doing really coordinated, awesome actions, and that's what we call biological life. Mostly structured around carbon, which is the organic molecule. Okay, so in here we're going to identify structures and components of prokaryotic and eukaryotic cells, which is building on our last lesson. We're going to identify and distinguish structures and functions of common macromolecules used by biological systems. We're going to describe the concepts of atoms, molecules, chemical bonding, pH, and the polar nature of water as they apply to the structure and function of biological molecules. So we're going to cover a lot of different topics here, going into water as this main universal solvent. That's when we'll get into polar nature of water. That's a pretty interesting topic. Okay, so let's dive in. So the components of cellular structures, as we read through this, you want to select carbohydrates, lipids, nucleic acids, and proteins. We saw those all in our molecules from our last lesson. Carbohydrates, that was like glucose that we broke down. Lipids we found in the cell membrane. And nucleic acids we found in the nucleus when you went in there to grab your RNA. That's a type of nucleic acid, as well as DNA. And then we made a protein. And those are the four big molecules that make up the cell. All right, next. Atoms and bonds. Shown to the right are diagrams of each of the components you identified in your exploration. They, in turn, are made up of smaller pieces of atoms connected by things called bonds. Identify the graphic below in which pieces you think are atoms and which you think are bonds. So this connection, this tube here, that is a bond, and the actual circle is the atom. Atoms and bonds are often represented in chemical structures by balls and sticks, but like organelles are more complex than they are often represented. Later, you'll learn about bonds. For now, you'll explore what an atom is. In 30 to 140 characters, give your own definition of an atom. So here I wrote an indivisible unit made of protons, neutrons, and electrons. But you want to come up with, what do you already know about atoms? I'm sure you've learned about it. So from your head, come up with something between 30 and 140 characters. So here we're going to... Are we recording? Small technical difficulties. Let's see. Hold on. All right, so we're recording now. Sorry about that. Just figuring out how this program works. Okay, so the plum pudding model, where does this idea come from? From the fact that this circle with little negative particles inside of it looks like, in Britain, they would have plum pudding cookies. You might call this the chocolate chip cookie model, if that is your preferred cookie. But basically the idea is a beam of light was sent out from this cathode ray which sounds very sci-fi-y, but it kind of is. So it was a beam of particles. It hit some coils, and the coils deflected a positive charge. And so because there was a negative, because it moved the opposite way, it moves towards the positive field with a negative charge because opposites attract, right? So this beam of particles moved towards the positive end, having that then was evidence to say, okay, there must be charged particles within an atom. And so we created this model, the plum pudding model. So what was J.J. Thomson's biggest difference from Democritus's model? Negative particles, atoms broken into smaller pieces, positively charged atom, the term atomos, or the use of the cathode ray tube. And here the question's asking for the biggest difference. So I would say negative particles. But that's not right. Can there be another? It's not the biggest difference. So remember that Democritus said atoms couldn't be broken into smaller pieces. And so here that's probably the biggest difference. Dun, 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 well done. So yeah, Democritus couldn't even conceive of this because he didn't have a cathode ray tube. He was just using a thought experiment to figure out what an atom was. All right, enter this man. His name is Rutherford. He's one of Thomson's students, so you can see how ideas get built upon each other. As one person dives deeper into what we currently know, that sort of paves the way for more people to understand what is going on and build upon that knowledge. So Ernest Rutherford took his experiments another step forward, looking at positive particles instead of negative particles. Thomson's models predicted that atoms were not very dense, especially compared to alpha particles. To test this, Rutherford set up a piece of gold foil made of gold atoms in the middle of a ring of material that could detect small, dense, positive particles called alpha particles. He then shot a beam of alpha particles at the gold foil. Which scenario do you think is most likely for this experiment's results? Make a hypothesis. All right, so we have the beam of alpha particles will pass through the foil unchanged. The beam of alpha particles will bounce off the foil back the way it came. The beam of alpha particles will deflect randomly off the foil. The beam of alpha particles will pass through mostly unchanged and some will deflect randomly. The beam of alpha particles will pass through mostly unchanged and some will bounce back. Or the beam of alpha particles will stick to the negatively charged atoms. And so here, if you imagine, we're kind of getting to see here, if you have a atom that has some positive molecules or particles, let's say, on the inside, and it hits something else that has some other positive, that's like mostly positive particles, what do you think would happen? So we can just try a couple different ones. I think this is probably what's going to happen here. The beam of alpha particles will pass through mostly unchanged and some will deflect randomly. So let's go ahead and see what he found. He found that to his surprise, most particles went through, but some were deflected or even bounced back. Why did this surprise Rutherford? Thomson's model of the atom was all negative, so positive particles should have stuck. Thomson's model of the atom was neutral, so it should not have interacted with all the alpha particles. Thomson's model of the atom was all positive, so it should have bounced all the particles back. Thomson's model of the atom had a diffuse negative charge, so the particles should have moved in circles around it. Thomson's model of the atom had a diffuse positive charge, so the particles should not have been deflected very far. So we know Thomson's model of the atom did account for having positive and negative charges, so we can rule out number one. Rule out number two. Rule out number three. The moving around in circles is a little bit of an odd statement there. I'm going to go with the diffuse positive charge. So the particles should not have deflected very far. And it says correct. Thomson proposed a diffuse positive charge, so seeing the fast, heavy, dense alpha particles deflected so much was like firing a bullet at a sheet of paper and seeing it bounce off. Yeah, so imagine you had, so this kind of points at that there's a central, dense, centralized concentration of all the positive particles, and you might know this as the nucleus. Okay, so we're getting closer to the common understanding of what an atom is, how it's structured. We redefined it even further. Rutherford reasoned that if fast, dense, positive alpha particles were being heavily deflected or even bounced back, they must have been hitting something else which was positive and dense. He calls this dense positive region the nucleus. So here now we have negative electrons on the outside, mostly empty space for everything else. All right, so now how was Rutherford's model different from his mentors? Rutherford's model had a small, dense positive nucleus. Rutherford's model had a large, diffuse positive region. Rutherford's model had a small, negative particles called electrons. So we know that Thomson's model also had electrons, so that's probably not right. Rutherford's model had positive particles called protons. He hasn't quite called them protons, right? Rutherford's model had small neutral particles called neutrons, and we definitely don't know yet that there are neutrons, so let's go with the small, dense positive nucleus. And that is correct. All right. So now we're getting to a little bit later in the timeline, and another student, the next generation, called Rutherford. After Rutherford is Niels Bohr. He's responsible for the most common representation of the atom today. You've probably seen this symbol used more than once for atoms or chemistry, so if you think of like Jimmy Neutron, that's what I think of this model. And Bohr learned that electrons have specific energy and that the electron's energy limits where it can be found in the atom. Think of it like a staircase. You can be on a step, but never between steps unless you're moving from one step to the next. His model, the Bohr or planetary model, puts this electron in orbits around the nucleus based on their energy. All right, so now we're selecting all that apply compared to Rutherford's model. Distinct energy levels for electrons, yes. Electrons are outside the nucleus. Well, Rutherford thought that as well. The nucleus is present. Rutherford thought that as well. Distinct orbits for electrons, correct. And distinct colors for electrons. That is true, but I wonder if Bohr knew that. And I don't think he did, so let's check that out. Excellent. All right, and then we get to Schrödinger's atom, which gets even more complicated, but closer to the more accurate version of an atom. So Bohr's model was still missing something, needing some refining. Erwin Schrödinger, which you might know as from Schrödinger's cat thought experiment, he was an Austrian physicist, performed complex calculations that revealed electrons in an atom don't travel in orbits, but rather randomly in three-dimensional space, in which he called clouds. Like orbits, these clouds are based on the energy of each electron, but are larger and less predictable. So how is Schrödinger's model different from Bohr's? It includes protons that are positive and dense, so Bohr's did as well. It has electrons that are negative, same with Bohr's. It includes neutrons, and here we still haven't really touched on neutrons, so how about this one? Electrons don't have orbits, but have volumes they may occupy. It sounds pretty accurate to what we just learned. Electrons don't have energies, but have volumes they may occupy. Okay, so Schrödinger took the idea from Bohr, turned orbits into clouds, because we don't quite know exactly where they may be, so we can't predict them along orbits. All right, so where do we get to neutrons? we have to rely on these experiments by James Chadwick and others in the early 20th century. They discovered that the nucleus was not one big dense area, but rather made of two particles, one positive and one neutral, with equal masses. These particles were named protons and neutrons. At this same time, experiments also revealed that the electron has far less mass than the proton and neutron. It would take more than 1,000 electrons to equal the mass of just one proton. If atoms were a football field, the nucleus would be the tip of a pencil on the 50-yard line. So that really puts it in perspective for you how much space is actually in an atom, which is crazy. Where is most of the mass in an atom located? So is that the nucleus or the electron cloud? That would be the nucleus. What makes up most of the volume of an atom? And that's mostly empty space. Are all of the pieces are in place, or are they? All of the research from Democritus through Chadwick and beyond has led to the modern model of the atom, called the quantum mechanical model. It has three parts: a proton, with a +1 charge and a mass of 1 atomic mass unit located in the nucleus; neutrons with no charge, a mass of 1 atomic mass unit located in the nucleus; electrons with a -1 charge, a mass so small it will be 0, and located in electron clouds. And so we're going to continue to look at the model. Adding in particles called quarks are the subject of current research all over the world. Shown at the right is a diagram of quarks that makes up a proton. Okay, and we're going to pause. All right, and so now we have an opportunity to match what we just learned, connect the name of the scientist to their contribution to the modern model of the atomic structure. So what we do here is we connect the two. So I would connect Bohr to, what did he have? The initial concept of protons and neutrons. Did he talk about electrons in clouds? Electrons in orbits at energy levels. I would connect it like that. Then I take Rutherford. He had the gold foil, which allowed us to say that there's a dense amount of particles in the nucleus, which is the positive protons. There we go. Thomson had our plum pudding model, and that taught us that we had electrons. So I want to connect these two, clicking on that, clicking over there. Schrödinger then said that we can't really predict where any of those electrons are going to be, so let's say that they might be in clouds. Chadwick added in the idea of neutrons, and Democritus from Greece said that we had the initial concept of an atom being indivisible. Let's check that out. Did we do it? Dun, 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 woohoo! Each scientist made an important contribution to our current understanding of the atom. All right, so here we have another chance to practice what we just learned. We want to fill out an electron. What kind of charge does that have? It's negative. It has a very low mass, and the location is, we would say, in a cloud. That's our most recent understanding. And then where we have protons in the nucleus with a +1 charge, the mass 1 atomic mass unit in the nucleus. And here we have a neutron with a neutral charge, mass still 1 atomic mass unit, located in the nucleus. There we go. All right, so now you have a lot more information about what an atom is. Go ahead and redefine your definition of an atom. And maybe now you want to talk about the location of electrons, how they orbit in clouds. You might want to talk about protons and where they're located in the nucleus. You might want to talk about neutrons, also in the nucleus. with an atomic mass of 1. And then you maybe want to talk about how the development of atomic structure reveals that there are actually subparticles, subatomic particles that make up the concept of an atom. Something like that. Put that in your own words, and that will get you points. Whoa there. Okay, it's important to be concise. Shorten your definition a bit. So I gave you way too much information. We went over that amount, so there we go. All right, so we're going to dive in deeper to what it means to build up an atom, protons, neutrons, and electrons. All right, so here we're getting deeper into the understanding of atoms. Now knowing that they can have different numbers of neutrons, remember our neutrally charged particles within the nucleus that have atomic mass, and we call those isotopes. Isotopes have different behaviors. Some might be used faster in chemical reactions or be radioactive. So here we have, I think this is supposed to indicate uranium, which is a radioactive molecule, atom. Atoms of an element that have different numbers of electrons are called ions. Okay, so we have two different concepts here. Isotopes, different number of neutrons. Ions, different number of electrons. So which one would change the charge? That's the question. If you have ions and electrons are charged, ions are going to have a different number of charge, a different charge. Normally atoms that have equal number of protons and electrons are neutral, right? The positive and negative come together to neutralize the atom. But when it's charged, when it's changed, the atom gains a charge, either positive or negative. Here we go. Let's dive in. All right, so there's a couple different ways we can represent molecules. To this point, large molecules have represented as schematics, either as ribbons, balls, and sticks, or as large amorphous blobs. As you found out in the previous screen, that these schematics are ways of representing the various atoms and bonds in each molecule. Often it is useful to represent which particular kind of atom is at each place in that model. To do so, we use chemical symbols that are unique to each element. C for carbon, O for oxygen, H for hydrogen, and so on. So here we have ball and stick and a structure. This is what's called a Lewis dot structure because it shows the electrons on the outside and the bonds with lines. Here the bonds are the tubes and the atoms are the balls. And here the atoms actually have a representation with a letter. So you can read from this that water, which is H2O, has 2 hydrogens and 1 oxygen. And here's something called methane, has 1 carbon in the middle and 4 hydrogens on every side. You can see that over here in the structure too. So the question asked is what information is provided by the ball and stick model and chemical structure for each molecule that is not provided in the formula. So here's the formula, would be on the left side. So we get the chemical name, how the atom of each element are connected, that you don't see in the formula. How many atoms, we can tell that. We can tell H2O, we can count up the number of elements, or atoms in each element. How many atoms of each element are in the molecule, we still know that, yep. Both the formula and the model structure provide the same information. We know that's not true. Which elements are in the molecule, we can still see that from the formula, so that is our best bet. That is correct. All right, using what we learned in the chemical structures on the last screen, which element do you see in these 4 molecules? Enter your answer using the atomic symbol shown in the diagram. Do not use spaces or commas. Okay. So I see a C. I see an H. I see an O. I see an S. I see an N. And here down here, I see a P. Next. All right. So this makes up all of the elements that make up most of life. Sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. They're essential to life, and they're part of over 100 more that are, okay, so then there's 100 more that are used in the periodic table of elements. We can create atomic bonds. Ionic bonds are when two or more atoms bond with each other. They do so by sharing or transferring a part of themselves called an electron, a negatively charged particle. One type of bond where an electron is transferred between atoms is called an ionic bond. Ionic bonds are fairly rare in biology but common in geology. They tend to form between elements which are far apart on the periodic table. So if I take something like sodium and chloride, that's atomic number 11, atomic number 17, we've seen that forms a salt, and a salt is formed by an ionic bond. Sodium actually gives up an electron, and the chlorine gains that electron, and that makes them actually neutral when they're together. It's important to note that H is unusual and often acts like it is above with F. Which of the following compounds is likely to have an ionic bond? Here we just said salt. And that is correct. All right, we have another type of bond where electron is shared between atoms. It's like a covalent bond. It's called a covalent bond. Covalent bonds are found in many molecules, especially those used in biology, and tend to occur between elements close to each other in the periodic table. So here we can, if you see C, P, O, H. H is over here, but like they just said, it tends to act like F. And N. They're all close to each other. And so they do something called covalent bonding, which is sharing their electrons. All right, so let's get into our macromolecules. We have our nucleic acid, that's like DNA and RNA. When two or more atoms bond together, we call the resulting structure a molecule. Some molecules, called polymers, are built on similar repeating units called monomers. The four types of molecules below are an example of these polymers. Shown to the right is an example of a biological polymer, a nucleic acid, and below, candidates for the smaller units is built form. So here we have a molecule of DNA, and the repeating unit is this nucleotide. That's one of these structures. You can relate this hexagon with a pentagon next to it is like right there. And then this pentagon is like right there. And then it has this little backbone, which is this phosphate group that connects them. So that is the repeating unit. And if you look in, you can see this one looks pretty similar, but a little bit different. And we'll get into what that is. But that builds up the big long chain that is DNA. All right, here we have a lipid. It is made up of, this is the repeating unit. The repeating unit is a fatty acid. Here we have a carbohydrate, and the repeating carbohydrate is a monosaccharide. Saccharides are sugars, so monosaccharide is like one sugar. You might also have disaccharides, polysaccharides. So this is in a pentagon shape. It's hard to see, but that would be like the basis of glucose. You string a bunch of glucose together, you might make something called cellulose, which builds up the structure of a plant. Like when you eat celery, you're eating cellulose. And our last one here, this is a nucleic acid. This is a protein. This protein is made up of amino acids. So I think of this when bodybuilders go to the gym and they are getting their nutrition, sometimes they go, I need to get my aminos. If you get your aminos, you're building protein in your body. And this is just saying each polymer is made of different monomers. Proteins are made from amino acid, lipids, fatty acids, carbohydrates, monosaccharides, disaccharides. Nucleic acids are nucleotides. So here we have a chance to practice that. Amino acid, proteins. Their function is to speed up reactions, aid in movement, contribute to structure, and perform functions of the cell. Carbohydrates, do you remember the monomer would be a monosaccharide? They're used in energy storage and to assist in structure. If we put a bunch of nucleotides together, that makes up a nucleic acid. Their function is to store and transport information and speed up reactions. And lipids are made up of fatty acids, and they provide cell structure like the cell membrane, long-term energy storage like fats in your body. All right, here we can see our biopolymers in action. This should look familiar. This is a cell membrane. And in a cell membrane, we'll see a couple of the different types of different polymers. All right, so if we zoom in, it's asking us, what major differences did you notice between the bacterial cell and the animal or plant cell? What seemed similar between the bacterial cell and animal or plant cell? So, did they both have a membrane? Yes. Did they both have proteins? Yes. Did they both have membrane structure? Yeah. Did they both have internal structures or organelles? That was what was different. Remember, bacterial cells don't have organelles. They both have genetic material. They both have lipids. They did not both have a cell wall. Remember, animal cells don't have cell walls. They both had carbohydrates. They both had nucleic acid. Maybe it was hard to tell, but the size was not that similar. And they both had ribosomes. Okay, I guess they both have ribosomes, but the ribosomes are different. All right, so the major differences then we can see are the structures, the ribosome structure, the size, and whether or not they have a cell wall. All right, membrane components. Shown to the right is a diagram of a membrane or a thin layer separating two regions. Membranes are found in the cell as the cell membrane itself or as the membrane of a compartment within the cell. What biopolymer or macromolecule do you think the major component is? So this is made up of, these are fatty acids. Fatty acids make up lipids. All right, so let's take a deeper look at what lipids look like. I think they look like, they're sort of funny. They're this ball with these two tails. So the ball is a phosphate, so it makes up a phospholipid. How are these arranged in the membrane? Phospholipids are arranged with the phosphate head facing each other. So does that seem accurate? No. The phosphate is actually on the outside and the tails are facing each other. Oh, there we go. Phosphates are arranged with the hydrocarbon tail facing each other. So you have the tail. And that is because they both like to repel water. So they're going to go in and hide towards each other. Here it's saying the arrangement of lipids is due to a unique property of the phosphorus group and the water. Both molecules have covalent bonds. What type of bond is that? Remember, covalent is when they share their electrons. There we go. A bond where electrons are shared. A covalent bond of water is the head of a phospholipid, and they are shared unequally. That is, they spend more time around one atom in the bond, in this case oxygen, than the other. That makes them have one end that is a little bit negative and one that is a little bit positive, kind of like the magnet's north and south pole. So here that's what this little swirly symbol is. That means partially. So here we have the oxygen on one end, that's what's represented by the red, and it's partially negative because the electrons like to spend more time around the oxygen than they do the hydrogen. And that in turn makes the hydrogen more positive because it has less of the negative electron charge influencing it. And that sets up what we call a magnet. And it's like the poles, positive and negative poles, and we call it then polar. So what do you think that means about the interaction between phosphate and water? So here we have water at the head of a phospholipid attracts each other like magnets, so they interact well. That seems pretty good. Water at the head of a phospholipid attract each other like magnets, so they interact poorly. Means absolutely nothing. If you're a nihilist, you might want to choose that one. Water and the head of a phospholipid repel each other, so they interact poorly. Or water and the head of a phospholipid repel each other like magnets, so they interact well. So the best combination of ideas here is water and the head of a phospholipid attract each other like magnets, so they interact well. And here they're bringing up another term, hydrophilic. So hydro meaning water, and philic meaning loving. So we have a water-loving molecule due to their strong interaction with water. All right, so here, this interaction isn't necessarily a bond. It's not ionic or covalent, but it's called a hydrogen bond. It's a weaker attraction between nitrogen, oxygen, or fluorine atoms in a molecule where there is also an attraction to nitrogen, oxygen, or fluorine on a neighboring molecule. All right, so how do we define ionic bonds? How do we define hydrogen bonds compared to covalent and ionic bonds? Hydrogen bonds don't involve electrons being shared or transferred, that's true. Hydrogen bonds occur with other molecules instead of inside molecules, that's also true. Hydrogen bonds share hydrogens instead of electrons. It's maybe easy to think that, but that's not necessarily the definition. It's still having to do with an attraction created by electrons being shared via covalent bonds. They're not very strong, so that's not a good option. Hydrogen bonds only happen with water. Nope, they can happen with other molecules as well, like it was just saying up here, nitrogen, oxygen, or fluorine. And hydrogen bonds aren't different from covalent. They are different. We just learned that. Okay, so that was correct. Good job. All right, so now we're getting into this hydrophobic. So if hydrophilic meant water-loving, if you had a phobia, maybe you're afraid of something, hydrophobic means it's fearful or it does not like water. And here the tail of a phospholipid is hydrophobic. So it does not like to be by water. Do you think they would be polar like the phosphorus group? The tail would be polar like the phosphorus group. The tail would not be polar like the phosphorus group. So if the phosphate head interacts well with water because they're both charged, the tail is actually not charged, it's not polar, and therefore it does not interact well with water. And that's this next question. Do you think that they would interact with water as well as, better than, or less than the head group? The tail end will interact with water less than the head group. Okay, so here we have a chance to, again, show what we just learned. If A is the phosphorus head, it does like water, hydrophilic. B does not like water, hides from it, hydrophobic. C again is the head, likes water, hydrophilic. There we go. All right, so zooming back in on the cell membrane, we have 3 different other features to explore. So let's go and zoom in on the protein channel. Proteins provide a variety of tasks within the body, from speeding up chemical reactions to helping transport material between different areas. They are made up of monomeric units called amino acids. Monomeric meaning monomer, like the individual repeating units. A long chain of connected amino acids is called a polypeptide because they form a peptide bond, polypeptide. When the chain folds into the correct 3D structure, is now a functional protein. So here we have our individual monomers, amino acids. We make a polypeptide when we link them all together, and when they fold in a particular structure, that makes up our protein. All right. Now, let's look in at the protein structure. From amino acids to peptide chain to fully formed protein, various inter- and intramolecular forces are present. Inter meaning between, intra meaning within, between. So inter like between the molecule, and intra outside or within other molecules. So I don't know if you ever play intramural sports. That's like 2 different teams playing against each other from different schools. I guess intramurals would be within your school. Okay, in the image to the right, you can see all of the various intermolecular, so within the molecule forces. These forces can be disrupted and cause the protein to lose its shape. So within this molecule, you have van der Waals forces, that's keeping this group together. Remember our hydrogen bonds, that's a weak bond, but it keeps this ribbon sitting right there. You have ion-ion salt bridges, it's keeping this here. And then you have dipole-dipole interactions. So that's like 2 different charged particles. Again, this is all interactions between charges within molecules. How in the world would you know this? Okay, order the bonds below from strongest to weakest. And here's a hint, I guess this is how we know. Nonpolar interactions, also called van der Waals forces, are weaker than interactions between polar molecules. All right, so now we have van der Waals being weaker. Okay, so order from strongest to weakest. So the number 1 would be the strongest. Charged particles usually interact more evenly, even more strongly with polar molecules than other polar molecules do. So charged particles, that is going to be right there, ion-ion interactions. And then we have dipole-dipole interactions. And then hydrogen bonding, and we know van der Waals are even weaker than that. Oh wait, that was not right. Okay, so here we go. Remember, intermolecular forces such as hydrogen bonding, dipole-dipole, and van der Waals interactions are caused by unequal sharing of electrons that create areas called dipoles that are partially positive and partially negative. Van der Waals interactions are temporary dipoles. Hydrogen bonding is permanent dipoles and strong, while dipole-dipoles are permanent dipoles and moderate in strength. Okay, so here we have ion-ion interactions, hydrogen bonding, dipole-dipole, and then van der Waals. There we go. All those make the protein stable. All right, let's zoom in on this guy up here. All right, so moving on to carbohydrates. They help fuel your body and provide energy for everyday cellular functions. Carbohydrates are found in almost everything that animals digest and are used to store energy for later. They are made up of repeating units of sugar. These sugars can be used as energy storage, and as signaling complexes on proteins in the bilipid membrane. So we just saw that in the cell membrane, how there is this little molecule here. It can be used as a signaling molecule. For example, in your blood cells, on the outside of your blood cell, you're going to have your different blood types, and those are different proteins that stick out of the cell. Sorry, those are different. Oh, let's pause here. All right, just a little bit of research. There's both proteins and carbohydrates that make up the antigens that are on the outside of your blood. So that answers that. Back to the lesson. So select the elements that make up sugars. We have carbon, hydrogen, and oxygen. That's all that makes up a sugar. Okay, so when we zoom in on this carbohydrate, they have multiple functions within the body, and then we're just going to focus in on just one function. Attach to proteins or lipids and act as recognition and binding sites. This means that they can interact with certain molecules and form intermolecular forces strong enough to allow for a reaction, but weak enough to allow molecules to come and go. So that kind of makes sense. It's like kinetics, those little the fun toys that you can play with that link together and then you can separate them like a magnet. So looking at the monomer of the carbohydrate here, we have our monosaccharide. What type of intermolecular forces do you think would be strong enough to bond but weak enough to break apart when needed? So which one of these do you think it would be? Hydrogen bonds. And if we look at this molecule here, we might see some oxygens and hydrogens, so that might indicate to you that there would be a hydrogen bond. Okay, biopolymers in action. Let's continue on our lesson with biopolymers. Okay, so our final one here. Let's go and zoom into the nucleic acids. This is how genetic information is passed on from one generation to the next. In the cell, there exists a type of biological blueprint. It contains all of the information that allows life to continue. These blueprints are stored in deoxyribonucleic acid, or DNA. To accomplish this, the body uses another form of nucleic acids, ribonucleic acids, or RNA. Together, these two molecules allow all of our other biopolymers, including proteins, to be created and then used in the body. All right, so we're talking about the central dogma, or a main central theme in biology. It states that DNA transcribes RNA, which is translated into a protein. So this is the function of going from DNA, making a copy of it that can leave the nucleus, that we call that transcribing. Imagine if you were writing one book to another language, you would say you're transcribing that book. And then that is translated, so transcribing from one form to another form, and then translated into another language, which is a protein. All right, so let's take a look in at the structure. DNA is made up of a polar, remember that means that there's a separation of charge, phosphate, so there's a charge on the phosphate, and that's the backbone. There's a polar ribose sugar, so a carbohydrate, and a nonpolar nitrogenous base. Nitrogenous meaning it has nitrogen in it. These DNA strands then form helix structures in the body. That's this twisty structure. Why is it more stable for nonpolar bases to be in contact with each other than the surrounding water? Remember what it means to be nonpolar? So we might say something like nonpolar repels water. What about the charged phosphate group? So the charged phosphate group attracts water or interacts well with water. Why is it more stable for them to be in contact with water? The positive and negative poles interact well with the phosphate. The charged phosphate hen. Or here we might just say group. There we go. You may be on the right track. I don't think this algorithm actually knows what you're saying, but let's read what they have to say. Just so you're clear, the charged phosphate group interacts well with the polar molecules and are most stable on the outside of the molecule in contact with water. On the other hand, the non-polar parts of the bases do not interact well with water and are more stable within the molecule, shielded from the polar solvent, or the liquid outside. All right, so here we are. We have now explored how the 4 biopolymers work together in unison to control and regulate activities in and around the cell. Understanding that these 4 biopolymers are necessary for our cells to live, we know that they are common to all life on Earth. These 4 biopolymers represent, in many organisms, both what they ingest for nutrients and what they expel as waste. The presence of these biopolymers can provide evidence that life exists, or perhaps once existed, in another world. Okay, so if we're searching for signatures for life on other planets, with all the complex molecules and structures found in cells, which one of these do you think you should use in our search for life in the universe? Which molecules do you think come from and indicate the presence of life? Molecules? We got water, amino acids build up proteins, nucleotides build up nucleic acids. Hello. All right, monosaccharides make up carbohydrates. Nitrogen is responsible. There's fatty acids in our membrane, which is made up of lipids. Carbon dioxide is, I don't know, maybe, obviously we produce carbon dioxide as a byproduct. There's definitely hydrogen, and there's iron in our blood cells, but let's see what they think about that. What sorts of things do you think might complicate your search? Ability to analyze samples on other planets, sure. So the rover that goes to Mars, it actually picks up samples of rocks and analyzes what is present in those rocks. The size of the universe, there's so many different planets to search for, and they're looking for the Goldilocks conditions, temperature, amount of time around a star or a sun, ability to find these different molecules. Definitely your ability to send machines there. You could definitely contaminate it from Earth. Maybe you had something on your sampling equipment that had oxygen or water in it. It's hard to totally expel those. Non-biological ways to make the molecules you are looking for. Yeah, so you might have carbon dioxide. There's tons of carbon dioxide on Venus, but that doesn't necessarily mean there's life there. And not enough knowledge of how life came about on Earth, for sure. We only hypothesize about what happened in the past. And we have a lot of evidence, but we are always piecing that together. So let's see how we did. Okay, so we have sent several other probes to other planets. Okay, so we can do that. What substances like water, oxygen, nitrogen, carbon dioxide, hydrogen, and iron might be necessary for life? They are not produced by it. Think again more and look at question 1. Okay, so definitely, oops, we forgot oxygen. And let's do carbon dioxide. Okay, so yeah, thinking closer about this, sometimes it takes, just needs to, oh my gosh. Okay, so water, it's produced by life, but not, it's used by life, but not produced by it. So that definitely narrows down. We only want to be selecting our macromolecules, which makes a lot more sense here. There we go. So amino acids, proteins, nucleotides, nucleic acids, monosaccharides, carbohydrates, fatty acids, and lipids. We can analyze other samples, and we can send machines to collect those samples. Let's see how we did. Woo! Awesome, there you go. Next. Time to pause and reflect. We just spent some time learning about the following 3 major topics. How well do you understand them? So now, atomic structure. They're made up of electrons, protons, and neutrons. That determines the element it is. We know that there are ionic, covalent, and different types of bonds depending on charges, such as hydrogen bonds. There's 4 different types of macromolecules. We know a little bit more about their structure now. And polarity, different ways that, say in water, they might share unequally, and more of the electrons get shared over by the oxygen, meaning it's more negative. And that makes hydrogen bonds. Okay. So, as we wrap up, it says cells are made from organelles, which are made of biological macromolecules, lipids, carbohydrates, proteins, nucleic acids. Those macromolecules are made up of smaller components, which are made of atoms bound together by their electrons. Those atoms are made of smaller components still, protons, neutrons, and electrons, discovered by the tireless effort of scientists over thousands of years, and that's what we learned about. This combination of atoms into complex molecules results in the processes that drive biology and is a signature of life that we search for on other worlds. All right, and we are all done. I think maybe it was that last screen here, we lost a couple of points, but total you should have about 200 points. And it should auto-load into the gradebook. So you do not, for this lesson, need to go and make a Canvas entry. There's no text box for you to complete here. But I would go back and check. I know some students, if you just did this through BioBeyond, you might not have it auto-uploaded, in which case just send me an email or make a comment on Canvas, and I'll make sure your grade is entered accurately. Maybe take a screenshot first. Wait. Okay, so that's about it. Make sure you do not need to do anything in Canvas because this is an auxiliary lesson, so it will auto-load. But I would go back and check. If you didn't open it in the modules, it might not load for you. So just send me an email and I'll make sure your grade is updated. Great job today. It took about an hour to get through this lesson. And you have one other lesson, so you can check that out. I'll do a similar video for that a little bit later in the week. And we'll talk about all this, really looking at some examples of how we've found life on other planets and how we build up a human on our live lesson on Friday. So I'm excited to see you there, and I hope this video lesson helped you make your way through this lesson, the chemical basis of life.